Welcome to part two of the respiratory system of the human body. Last section we looked at some gross anatomy and some microscopic anatomy of this respiratory system. Here in part two we're going to look at some pressure and some volumes. The idea is that pressure is going to drive inspiration and expiration. So let's look into how pressure changes in this respiratory system. And so one idea with pressure is that lungs do not collapse, and that's because we're going to have negative pressure in an intrapleural space. Let's go ahead and take a look at this intrapleural space and how we might form it. So to back up a little bit, we're going to have pleural membranes. We're going to have delicate membranes that surround the outside of the lungs, and it's going to create a pleural cavity. So this pleura is a collection of serous type membranes. So in other words, the double-walled structure. The visceral pleura is going to adhere to the lung surface, whereas the parietal pleura lines the internal thoracic walls. It's also going to line the surface of the mediastinum and then also the surface of the diaphragm. So again, it's a double membrane system. So with this double membrane system, we create a pleural cavity. So this pleural cavity is located between that viscera and parietal serous membrane layers. Then we'll get a sort of oily, viscous serous fluid that's produced by the serous membrane. That's going to cover the surface within the pleural cavity. Let's look at some values here. We have what's called a transpulmonary pressure. Here it is shown in this diagram. Notice that the prefix trans means across, so we're going across the visceral pleura. We're going from the pleural cavity into the inside of the cavity of the lungs. So this transpulmonary pressure is going to be a difference between the intrapulmonary pressure and the intrapleural pressure. And it's this transpulmonary pressure that's going to keep air spaces for the lung open. So therefore, if we can increase transpulmonary pressure, the lung size can increase. So these lungs are actually quite delicate tissue. They simply follow with whatever the thoracic cage is doing at the time. Let's take a quick mentioning of two homeostatic imbalances. Let me try that again. Atelectasis and pneumothorax. Pneumothorax occurs when free air sort of gets into that pleural cavity, and that is going to affect the pressure. So we might get pneumothorax being introduced externally, so that could be from a penetrating injury, a wound to the chest, or that could also happen from the internal region as well. Also, pneumothorax is going to sometimes cause a collapse or a deflation of the lungs. This collapse or deflation is what we're talking about when we say atelectasis. The collapsed lung will remain deflated until the air has been removed from that pleural space. This slide is just a sort of general introduction to pulmonary ventilation. Pulmonary ventilation is just a snazzy way of saying breathing. So pulmonary ventilation is the movement of air between the atmosphere and the alveoli within the lungs. So inspiration brings air into the lungs. Expiration is going to force air out of the lungs. Let's take a look at this diagram on looker's right. We have a closed volume, and notice there's a piston, so this piston can adjust the volume. So on the left we have full volume, on right the piston has shoved down and become half of the volume. Let's take a look at these orangish yellow dots. These orangish yellow dots might represent oxygen molecules, carbon dioxide molecules, nitrogen molecules in the atmosphere, etc. The idea is when we reduce volume, we're going to increase the pressure. And that leads to Boyle's law. So Boyle's law relates to a movement of air in and out of the lungs. So movement in and out of the lungs is controlled by Boyle's law. And Boyle's law states that volume of a gas varies inversely with that of pressure. And this is all assuming that the temperature is going to be constant, because pressure affects this as well. So if we showed it in terms of an equation, we could get Boyle's law. Little typo there. Trying to write the mouse. Here's a mathematical way to express the relationship between pressure and volume between two cylinders. So again, Boyle's law states that at a constant temperature, the pressure of a gas decreases if the volume of the container increases. And so the idea is if we change volume, we can change pressure. And if we can change pressure, we can get a sort of air pressure gradient. And so this air pressure gradient is going to occur when the force per unit area is greater in one place than another. And that is how air generally moves in and out of the lungs. So when we talk about atmospheric pressure, the atmosphere is going to be the air and the environment that surrounds us. So atmospheric pressure is going to be a pressure that the gases in the air around us exert upon our lungs. And then we get a sort of collective volume of the alveoli. We call it the alveolar volume, and that's going to be associated with that intrapulmonary pressure. So speaking of pressure, how do we change the volume of this cage, this thoracic cage? We do it through a muscular activity. And so breathing requires skeletal muscle activity, and that's going to cause the chest size to change. So in other words, if we change the volume, we can change pressure. If we change pressure, we have air moving in and out. So generally speaking, these skeletal muscles of breathing are going to be classified into muscles for quiet breathing. Then we have muscles that are involved in forced inspiration, and then we have muscles that are involved in forced expiration. So let's start with muscles of quiet breathing, and that's going to be a diaphragm and the external intercostals. In this diagram, notice how diaphragm is moving. As the contraction of diaphragm occurs, that's going to increase the vertical dimension of the chest. Notice we also have the external intercostals working as well. So in other words, we make the chest cavity larger, but also by those external intercostals sort of contracting together, we sort of pull that chest cavity upward and outward, so to speak. So here's a nice diagram showing quiet inspiration. It's noteworthy that on looker's left of this diagram is showing muscles of inhalation, and on looker's right, we're looking at muscles of exhalation. So as we said, the muscles of quiet breathing are going to be diaphragm and the external intercostals. And then we have forced inspiration. Forced inspiration, that's going to use skeletal muscles like sternocleidomastoid, the scalenes, muscles like pectoralis minor, and erector spinae are all going to help with forced inspiration. Here we have in quiet expiration. Quiet expiration is going to be a passive process. There's a natural sort of elastic recoil that happens in the alveoli, and that's going to pull inward. So then when we forcefully exhale, a forced expiration is going to include muscles like internal intercostals. We're also going to use some abdominal muscles, and transversus thoracis is going to help us with that forced expiration. So in summary, it is skeletal muscles that control the movement of the thoracic cage. The thoracic cage can then influence the volume of the lungs, and the volume of the lungs affects the pressure, and the pressure differential, the gradient, affects how air comes moving in and out of those lungs in the thoracic cage. And again, I think that's all been summarized here in this diagram showing that process. Let's now turn our attention to factors that influence ventilation. We have three main factors: airway resistance, surface tension, and lung compliance. Let's look into each one of those. We say that airway resistance is going to be resistance to airflow, and that's going to depend upon the size of that airway. So let's look at this graph in Looker's right. We see that on the x-axis we have stages of branching. So take, for example, our main bronchi is in our first stage of branching, whereas those terminal bronchioles have had many branches by this point. On the y-axis we have resistance. So then we then see a sort of also a segment. There's going to be the conducting zone showing our earlier airways, and then the respiratory zone is going to be those alveoli and such. So in summary, on this graph we see how resistance increases as we move into the medium-sized bronchi, and then as we get away from this medium-sized bronchi, the resistance decreases. So this resistance can be altered, and it can be altered through elasticity of the chest wall. We can also alter the resistance through the change in bronchial diameter. And then in addition, collapse of the alveoli would increase resistance as well. So in summary, air resistance is one factor that influences ventilation, sometimes also called airflow. By the way, it's noteworthy of a homeostatic imbalance called asthma. Asthma is, one might get an asthma attack if those bronchioles become constricted. Another factor that affects ventilation is going to be alveolar surface tension. That is, how much surface tension is located inside those alveolar sacs. Earlier we talked about type 2 alveolar cells. We also talked about this respiratory distress syndrome of a premature infant. And the idea is that these type 2 cells, if they are not producing sufficient pulmonary surfactant, we're going to get an increase in surface tension. We might get an acute respiratory distress syndrome when these alveoli collapse upon one another. And again, it's because water wants to sort of bond with itself. A third influence on ventilation is going to be what we call compliance of the lung. We might define that as the sort of ease with which the lungs and the chest wall expand. And this is going to be determined by surface tension and then also the elasticity of the chest wall and the lung. Later on, in I think segment 3, we talk about some diseases that reduce compliance. For example, tuberculosis and pulmonary edema will reduce compliance of the lungs. So in summary, three main factors influence ventilation: airway resistance, alveolar surface tension, and compliance of the lungs. We now turn our attention to respiratory volumes. So earlier and today we've been talking about the process of moving air into and out of the lungs. And you might recall we referred to that as pulmonary ventilation. This term pulmonary ventilation might also refer to the amount of air that's inhaled in 1 minute. We might say the normal average adult breathes approximately 500 milliliters. That's half of a liter per breath. And that's going to occur about 12 times per minute. So we have a way to look at these volumes, and we have a way to measure these volumes. Here's a device called a spirometer. A spirometer is going to measure the volume of air that enters and leaves the lungs. So we'll be looking at this in our lab class. This diagram on looker's right is showing that half liter that we take in for inspiration and expiration. Let's take a look again at some of those volumes. We're going to be talking about these volumes in our laboratory class, but let's go ahead and run through these. This thing called tidal volume is going to be the amount of air inhaled and exhaled per breath during quiet breathing. So in the middle of this graph, these smaller waves, so to speak, is a normal quiet breathing. Well, let's say we were going to sort of take a big, deep breath inward. We might get an inspiratory reserve volume. The inspiratory reserve volume is going to be the amount of air that can forcibly be inhaled beyond that basic tidal volume. And again, this is also going to be a measure of lung compliance as well. So if we forced ourselves to inhale, you might notice that we can also force ourselves to exhale. So therefore, we're also going to get an expiratory reserve volume. By the way, this is a unique graph where it's starting over here. Typically, we process left to right. For a variety of reasons, this graph is processing right to left. So we talked about a normal breath, a big inhale, and then a big forced exhale. So this expiratory reserve volume is going to be the amount that can forcibly be exhaled beyond that tidal volume. That's also going to be a measure of chest wall and lung elasticity. Well, even though we breathe in really hard and we breathe out really hard, there's still going to be some air left inside those lungs. So we say that residual volume is going to be the amount of air left in the lungs even after the most forceful expiration. So from using those four basic volumes, we can then do what's called calculating capacities. And again, we'll talk a little bit more about that in lab class. We talk about things like inspiratory capacity. That's going to be the sum of the tidal volume plus the inspiratory reserve volume. When we talk about functional residual capacity, that's going to be the sum of the expiratory reserve volume plus the residual volume. And then we get to vital capacity. That's going to be the sum of tidal volume plus inspiratory and expiratory reserve volumes. In other words, that's going to be a nice deep breath in and a nice deep breath out. And then lastly, if we toss in a residual volume along with that, we get total lung capacity. Total lung capacity is the sum of all the volumes, including the residual volume. So now that we've talked about normal basic breathing, let's see how we might change some patterns in breathing. So when we say eupnea, we're talking about normal variation in breathing rate and depth. For example, my lectures during talking right now, my breathing rate is varying slightly. Well, we might have something called apnea. Apnea is going to be the absence of breathing. And this apnea can be voluntary, such as holding one's breath, or it can also be drug-induced or possibly the result of a neurologic disease. Similarly, later on we'll talk about sleep apnea. Sleep apnea is going to be a temporary stop of breathing during sleep. We also have the other different general types of breathing patterns. Tachypnea is it going to be rapid breathing rate, and we have costal breathing, which is a combination of patterns using intercostal and external costal muscles. Typically during exercise, we're going to need increased ventilation. We also have a diaphragmic breathing. That's by contracting and relaxing the diaphragm to change the lung volume. Some people talk about singing with your diaphragm. Perhaps this is what they're talking about. And then also, we can modify our respiratory movements to express emotions and also clear our air passageways. Well, let's go ahead and switch gears. We're getting out of pressure, and now we're going to look at gas exchange. So when we say gas exchange, we're talking about the movement of respiratory gases between the blood and the alveoli, or from the blood and systemic tissues. So we have three basic steps. We have ventilation, which we've already talked about. Then we have external respiration, or exchange of gases into the lungs. And then we have internal respiration. That's going to be exchange of gases within the tissues. So in this diagram, we'll start at the top. Keep in mind, here's the atmospheric pressure. Oxygen comes in. We're loading oxygen into this bloodstream. Notice this diagram is showing it red, suggesting that we have oxygen-rich blood. Recall that this is coming into the left side of the heart, and then we're going to give it a nice big strong pump from the left ventricle, and that's going to send that blood to all the tissues. We're going to perfuse the tissues. So here we've arrived now at the systemic capillaries. That means we're dumping off that oxygen onto those cells. We're doing things like delivering oxygen to cells in the brain so we can think. Delivering oxygen to cells like muscle cells so that we can move. Well, those cells do create waste gases. In biology class we talk about respiration, a different type of respiration occurring, cellular respiration. And it just so happens in a microscopic cellular respiration, it turns out that carbon dioxide is released as a waste product. So carbon dioxide is then in these tissues. We pick up that carbon dioxide into the venous system. This venous structure is going to bring it back to the right atrium of the heart, and then right ventricle is going to pump that oxygen-poor blood back up to the lungs, where we are going to dump off that carbon dioxide and then repeat the cycle as we pick up the oxygen. So let's look into some of those processes that are occurring. So one might ask, what forces or what influences might cause an exchange of gases, a sort of switcheroo of gases, so to speak? Well, we have three things. We have the composition of the air. We have how soluble that air is. That's going to be described by Henry's law. And then also the structure of the hemoglobin and its affinity towards oxygen and carbon dioxide. So let's start out with the composition of the air, and we look at something called Dalton's law. So before we start with Dalton's law, we talk about atmospheric air. A little fun fact for the day is that the atmospheric air is not mostly made up of oxygen. It's mostly made up of nitrogen. If the air was 100% oxygen, the entire world would explode upon one spark or strike of a match. So each gas in a mixture of gases exerts its own pressure. It exerts a partial pressure. So we see in this diagram, we can sort of break down the total of the pressure of each of these gases. So then we add them all up, we get a sort of sum pressure, a total pressure. So when we say this term partial pressure, that's the pressure exerted by each gas within a mixture of gases. And just like blood pressure, we're going to measure that in millimeters of mercury. We typically write this as a P followed with a symbol for the gas. So here's what we're trying to see, a partial pressure of oxygen here. So a subscript is going to tell us what gas we're talking about. So yeah, this atmospheric pressure is going to be the total pressure of all those gases combined. And that brings us to an idea called Dalton's law. And then Dalton's law states that the sum of all the partial pressures is equal to total atmospheric pressure. So therefore we get what's called a partial pressure gradient. And this partial pressure gradient exists when the partial pressure for a gas is higher in one region of the respiratory system than in another. So in other words, that's how the gases are going to move from blood to tissue or tissue to blood. So the main partial pressures we talk about in understanding the respiratory system is going to be the partial pressure of oxygen and the partial pressure of carbon dioxide. So here we have a view showing the connection with that partial pressure. So let's go ahead and start on the upper right. Here we go. So we're here in the alveoli of the lungs. Here we see the partial pressure of oxygen is high, and here in these alveoli spaces the partial pressure of CO2 is low. That's going to pretty much stay the same as we move through pulmonary veins and stay quite similar through systemic arteries. And then we arrive at the tissues. And again, remember these tissues are going through a biological process called cellular respiration. Again, that's quite different from the respiration we're talking about, but it is connected. The cellular respiration is producing gases. This cellular respiration is bringing in oxygen, and it's making CO2 as a byproduct. So we see in this diagram now the partial pressure of carbon dioxide is greater than 45 millimeters of mercury. So in other words, the partial pressure of carbon dioxide now is greater than oxygen. Notice how this level stays the same with systemic veins, and then also moving up into pulmonary arteries, we see that the CO2 level is still greater. Now we arrive at inspired air, this air that's around the outside, the external environment. That is going to be a large amount of partial pressure of oxygen and a very small amount of carbon dioxide. Then we complete the cycle over once again. And the next graph shows that, well, even though we expire and breathe outward, there's still going to be a little bit of carbon dioxide that remains within those spaces. So in this inspiration, this breathing in, what's happening in the alveoli of the lungs is a combination of the inspired air and then what is left over after the breathing, the pulmonary ventilation. So in summary, it's these gradients. It's going to be this partial pressure gradient that's going to promote movement of gas from blood to tissue and tissue to blood. Well, we're on this topic of what is going to influence the exchange of gases. We've talked about Dalton's law of partial pressure, and now we move into gas solubility. In other words, Henry's pressure. Henry's law. So Henry's law states that at a given temperature, the solubility of gas in a liquid is dependent upon the partial pressure of the gas in the air, and then also the solubility coefficient of the gas in the liquid. So when we're talking about a solubility coefficient, we're talking about the volume of gas that dissolves in a specified volume of liquid at a given temperature and pressure. So let's take, for example, scuba diving, water diving, so to speak. Some people might think those air tanks are made up of entirely oxygen. That's not the case. that those air tanks have nitrogen then in there as well. So the nitrogen has low solubility, the CO2 has high solubility, and the oxygen has relatively low solubility. This idea also affects the hyperbaric chambers as well. So sometimes people talk about compression sickness, moving up to the atmosphere too quickly. So the idea, the main idea here is that carbon dioxide is more soluble in oxygen, and that is going to be more soluble than nitrogen. So then what happens then is gases with low solubility coefficients require larger pressure gradients to quote-unquote push that gas into the liquid. Let's go a little bit more in depth into Henry's law and how the rate of diffusion of those gases might be affected. One way we might affect that rate of diffusion is the concentration gradient. So the greater the difference, the faster that rate of diffusion. In addition, surface area. The greater the surface area, the greater the amount of diffusion. So these lungs have a large amount of surface area, a whopping 750 square feet of alveoli surface area. That greater surface area is going to increase our rate of diffusion. Also, diffusion distance. Buildup of fluid in the lungs is going to increase the diffusion distance, and that can affect the partial pressure. And then lastly, molecular weight of the individual molecules. CO2 is much more soluble in water than oxygen is, and that's going to allow CO2 to diffuse a little bit faster. So in summary of the things we've talked about, oxygen is going to diffuse across the respiratory membrane from the alveoli into those capillaries because the partial pressure of oxygen in the blood is lower than the partial pressure of oxygen in the alveoli. And then carbon dioxide diffuses across that respiratory membrane from capillaries into the alveoli because the partial pressure of carbon dioxide in the blood is higher than the partial pressure of carbon dioxide in the alveoli. So this efficiency, how efficient is this diffusion of oxygen and carbon dioxide? That's going to be dependent upon those factors we just talked about, the surface area and the molecular weight and so on. So that then leads to an idea called ventilation perfusion coupling. And this could generally be described as the ability of bronchioles to regulate airflow and also arterioles to simultaneously regulate blood flow. So when we say ventilation, ventilation is going to be altered by changes in bronchodilation and bronchoconstriction. And then perfusion is going to be altered by changes in pulmonary arterial dilation and then arterial constriction as well. So again, we have a sort of homeostatic balance matching ventilation and perfusion of that tissue. So we're still on this topic. We're talking about how exchange of gases can be influenced. And our third and final influence is going to be the structure of hemoglobin and its affinity or liking, so to speak, for oxygen and carbon dioxide. So when we say gas transport, we're talking about the movement of respiratory gases within the blood between the lungs and those systemic cells. And we've been talking about how oxygen gets transported. And again, as a review, the ability of blood to transport oxygen is dependent upon that solubility coefficient of oxygen in the blood plasma and the presence of hemoglobin. It is noteworthy that 98% of that oxygen is going to be carried by a hemoglobin molecule. And we have a diagram on the upper left showing that in one blood vessel, there's many red blood cells flowing through. If we look closer inside one red blood cell, we're going to see several hundred hemoglobin molecules. And it is these hemoglobin molecules which transport oxygen. The oxygen binds to a heme group on that hemoglobin molecule. So since the solubility coefficient of oxygen is very low, most of the oxygen in the blood must be carried by hemoglobin in the erythrocytes. It is noteworthy that hemoglobin also carries carbon dioxide, about 20% of it, but most of that is going to be converted by red blood cells with carbonic anhydrase to form bicarbonate ions. So let's look a little bit closer now how carbon dioxide gets transported. So here is an extremely zoomed in view. Notice we have the tissue cells that are producing waste products. They're producing carbon dioxide as a waste. And notice on looker's right, we have the blood vessel with a red blood cell floating through that. So carbon dioxide gets transported in the blood from the systemic cells to the alveoli through dissolving in the plasma, but it also is going to attach to the globin portion of the hemoglobin and also as a bicarbonate dissolved in plasma. 70% of the carbon dioxide transported in blood diffuses into erythrocytes to form this bicarbonate. And so then when blood reaches the pulmonary capillaries, carbon dioxide is then regenerated from that bicarbonate. So in summary, hemoglobin has a certain structure, and the structure of that hemoglobin lends to a certain affinity for oxygen and carbon dioxide. So again, this hemoglobin transport oxygen, it's going to be attached to an iron, and then this carbon dioxide is going to be attached to a globin. And the binding or the releasing of the oxygen or the releasing or binding of the carbon dioxide is going to cause an actual deformation, a conformational change in that hemoglobin, and that's going to temporarily alter the ability of hemoglobin to bind another one of those substances. So we say the amount of oxygen bound to hemoglobin is expressed as the percent of oxygen saturation, therefore, of that hemoglobin. So as the partial pressure of oxygen increases, the hemoglobin saturation increases. So let's take a look at this diagram. On the x-axis, we're going to see partial pressure of oxygen in terms of mercury, millimeters of mercury. And on the y-axis, we see percent saturation of hemoglobin. So notice lower level, 0 molecules of oxygen attached, 1 molecule, 2, 3, 4, and 4 is going to be our maxed out amount. So we see a sort of relationship, a sort of curve that relates partial pressure and saturation of the hemoglobin molecule. And so this, as we can see, this oxygen hemoglobin saturation curve is a graph that's going to relate a partial pressure of oxygen and the percent oxygen saturation. So what this curve is showing then is basically a change in partial pressure from 20 millimeters of mercury up to 40 millimeters of mercury results in a large increase in hemoglobin saturation. Then progressive increases in that partial pressure of oxygen cause less dramatic increases in that hemoglobin saturation. And we can see that as the graph sort of levels out there towards the end. Well, that does it for part 2 of the respiratory system. Stay tuned for part 3 coming up here soon. That's where we talk about how we control respiration, the nervous mechanisms, and then also some homeostatic imbalances.